All right. So our next speaker is Adheeba Ali from Uday's lab here. He's here in support. Adheeba, are you able to share your screen? Right.
Hi,
Adheeba,
sir.
go ahead and take it away.
Hello, everyone. So we have done our work on microplastics exposure in human liver. So we are going to present our topic. So as Miss Emma said, we don't need an introduction over the microplastics and what the exposure of humans to the microplastics. So what happens? Like, let us take a look of what happens when microplastics like get into the humans, or these microplastic cannot like cross the biological barriers and accumulate in organs, and does it contribute to disease. So like while looking into the microplastics exposure to the human organs, the liver is organ that is most likely to answer our question. So it receives 75% of the blood supply from the portal vein, and it drains directly from the GI tract. And so if microplastic cross the gut epithelium, the liver is their first major destination. So there are uh, number of studies like the preclinical and cell line data that has been implicating that microplastic can cause the oxidative stress, mitochondrial dysfunction, inflammatory cytokines and release, then fibrosis, lipid dysregulation, and apoptosis. So certainly, our question was like, do the microplastic exposure to the liver is specific to the disease phenotypes? Do metabolic diseased liver accumulate more microplastic than the chronic liver diseases? Does the burden differ across the anatomical regions of the liver? Which polymers actually dominate in human liver tissue, chemically confirmed rather than assumed? So our research question was like, among the adults like undergoing hepatic surgery, is the intrahepatic microplastic burden assessed by the particle size concentration, polymer composition, and regional distribution is significantly greater in MASLD-associated liver disease, cirrhosis, and SCC than in non-MASLD chronic liver disease. So this was built on the like this hepatic microplastic burden will be significantly higher in, like we hypothesis, it will be significantly higher in patients with massive associated liver disease than in patients with non-massive chronic liver disease. And reflecting the enhanced gut permeability, water transfer. Uh, so our primary objective was to investigate the presence distribution of microplastic in liver tissue obtained from patients with MASLD-induced cirrhosis, HCC, and non-MASLD chronic liver disease. And secondary objectives include to compare the presence and characteristic of the microplastic in liver tissue from patients, and to evaluate the intrahepatic regional distribution, and was to develop a contamination controlled digestion protocol and analytical protocol for the detection of microplastic in liver tissues. So we have included patients of greater than 18 years with disease, the above mentioned disease, which were undergoing liver transplantation, and the availability of enough tissue for the microplastic detection. And we have obtained IEC, and we had excluded patients with prior liver transplantation and the samples with procedural contamination risk. So after like we had collected the patients with liver mast cirrhosis and mast HCC, and alcohol related HCC, alcohol related disease, and after collecting the tissue under contamination control protocol, we had undergone sample digestion, and we like detected using fluorescence microscopy. Then we have used micro Ramen spectroscopy, Micro-Ramen spectroscopy, micro-Ramen spectroscopy for the quantification of microplastics, and for some IDEX, like for the morphology and for the polymer particle identification. So we had taken the liver tissue under the contamination protocol, and we used potassium hydroxide and sodium hypochlorite under 50 degree, 120 hours like incubation under 100 RPM rotation. Then we like defatted the, like, as the liver tissue is highly rich with fat, we defatted with the isopropyl alcohol and with the help of 220, and were underwent incubation at 50 degree and 50 rpm, and then it would underwent like the filtration using silver membrane. The silver membrane is particularly helping with the less interference with the microramen, so we have used the silver membrane. Then it went underwent the Nile red staining. The Nile red, it's a hydrophobic, like that it will help with the fluorescence, and the fluorescence microscopy we have done, then undergone with the Raman spectroscopy. Then we have selectively done the subsets of SEM and SEM EDS. These were the baseline clinical characteristics, and let's go to the results. So basically, so we can go to the next. So microplastic burden while analyzing across the different regions. So all of the most of the sample, that is 83 percentage of the samples, we got the presence of the microplastics. That mast cirrhosis we have mast cirrhosis we have like the median of 2.39, and the mast like HCC we had the highest median of microplastic. And this hepatic microplastic burden, it was higher in MASLD associated liver disease than comparing to the non-MASLD, which is mostly of alcohol-related etiology. And we had the limitation of small sample size. And by comparing the three groups, we had the highest median of microplastic burden in the MASLD-induced HCC. So from this slide, we can ask first, why would the mast livers accumulate more microplastic? This can we can answer this question with yeah, it can be because of the increased intestinal permeability, like mast disrupts the tight junctions proteins in the gut epithelium, allowing the larger particles to go through that would otherwise be excluded, and it have the direct portal delivery, like anything that crosses the gut wall is like funneled straight to the liver via the portal vein without the first passing through the systemic dilution. So this can be altogether give a picture to the result. And while correlating the BMI and like the clinical parameters with the microplastic burden, we have seen a positive correlation with the BMI and the microplastic burden. This it is particularly interesting because it can be read in like two ways. Obesity itself alters the gut permeability, microbiota composition, and inflammatory tone. So the higher BMI could mean the greater particle translocation, or alternatively, individuals with higher BMI often consume more processed and packaged food, which is well documented higher exposure diet. So higher BMI could just mean greater exposure. And while the previous slide. So intrahepatic regional distribution, we have like taken the sample from the three distinct regions like left lobe, right lobe, and periportal region. So we have compared the microplastic burden across these three regions, and periportal region showed the highest microplastic burden. This is this is where the periportal region is where the portal venous directly enters to the liver, and this can be a possible like reason to the higher concentration in the periportal region. And we have also like calculated the particle size and also evaluated the morphology. We can go to the next slide. So, like the particle size, we have got an average length of 31.6 micrometer particle size in length and median width of 9.2. We had ranged across 5 micrometer to 200 micrometer range particles were there. So the morphology, like most of the particles were of fragments, then fibers and films. We can go to next. The polymer composition, PET were the apart like unlike the other mostly seen polystyrene is seen, but we have got the PET polyethylene terephthalate and polypropylene in the highest concentration. We also found PVC, polystyrene, then polyamide and polyurethane. So we have done the micro Raman spectral analysis to confirm the particle identity. That thus we can confirm the particle identity based on the characteristics peaks. We can go to next. So these are the few Raman microscopic images that we have shown, and also the referral image, and also the what we have got the Raman spectra. So we have identified like the characteristic peak of the polyethylene terephthalate in the range of the characteristic peak of in the range 1725 and also in 1615 is also there. Then polypropylene also the characteristic peaks were present. Uh, so we have like the Raman microscopic image is also there, and also it is the image of like polyethylene. The characteristic peak of polyethylene was there. So, so we have analyzed, we have also analyzed initially we analyzed the fluorescence microscopy. These are the isolated particles that we have got. This most likely of fibers and fragments. And we have done the scanning electron microscopy to evaluate the microscopic, like morphological surface characteristics of the particles. So we have seen like the particles, like the chemically othered, like debrised particles, also the surface characteristics we have highlighted. The next slide. So we can go to next slide. Coming to like we have also done the energy dispersive X-ray spectroscopy. We have analyzed the elemental composition, and also while evaluating that, also we have uh, get the carbon oxygen composition at the highest concentration. That like pointing towards the uh, probable nature of the organic nature of the polymer. So while like analyzing the while coming to the conclusion, we have like found out 83% of the samples were positive with microplastics and periportal regions that demonstrated the highest microplastic burden. Then PET and polypropylene, it has shown a 78.6% of the dominant microplastics. And these all findings like support the gut-liver axis is a possible way that leads to the microplastic accumulation in liver. Um, so so our limitations we had several limitations of small sample size, and was a single center study. And we the Raman microscopy cannot access the nanoplastics, and also the environmental exposure histories, like we haven't got the data of the diets, occupational, or the environmental exposure based on the patients. So we're happy to be discuss further with any questions.
Okay. Adhiba, thank you so much. That was very nicely presented. Um, I don't see any questions in the chat right now, but I'll start with a thought. Have you considered complementary assessments for quantitation, either mass spectrometric like thermal desorption, GC mass spec, or pyrolysis GC mass spec? Or have you considered taking the histological sections and looking for either fluorescent or birefringent particles in your specimens to corroborate what you're seeing with the digested tissue?
Yes, sir. We are like planning to do the JCMS like further, and also the polarized imaging. Like it would be add to the result. Like we can compare to the result to the micro Raman uh, like confirmations. So we are planning to do that.
Okay. And how many samples did you ultimately obtain in this study?
We got like 12 patient sample that from three samples from each patients like 36 samples.
Okay. How many do you think would be adequate to overcome the power concerns that you have?
So yeah, we have to like get in each of more than 30 in each of the group in each phenotypes.
Okay. Okay. Cool. All right. Are there any other questions that people want to ask? You're welcome to just jump right in and unmute if you do.
Well, actually, this is the first study from India. Actually, we developed the like model. This is the like pilot study. Then we can have it some collaboration. Then we can like, move it to some like GCMA, some like further like transcriptome. So we generated some like pilot study. And then we can ahead with some like future directions. Like
Mhm.
thank you. Actually, like these also research is started from your paper. So like
Oh,
nature publication, then we think how to do it. Then she is the candidate. She has like done the like wonderful work. She is the first, and I gave the opportunity to her to represent. She had completed a PG the last week, like post graduation. She completed, so she is planning to do like PhD in. So we are like trying. So it's all like effort from our student and our lab. Thank you.
okay. Well, thank you very much. All right. I think we're at the end. Adiba, I really thank you for sharing your work. Uday as well for helping with this. This is
Thank
really
you.
nice to see. And of course, coming back to Emma's presentation, that was fantastic. Really great to see that advancing. And both of these questions, I think from my own research, the liver and the brain are really critical in both uh, accumulating, transferring, and potentially being targets of the micro and nanoplastics exposures. So really great to see these fields moving forward, and I appreciate everybody's contributions.